All right. So this is the second part of looking at RNA transcription and translation. We're actually going to look at translation. Um, we're going to be looking at the ribosome. Remember, the ribosome is made up of two parts. It's got the large unit and the small unit, which is RNA, ribosomal RNA, and proteins. In this ribosomal unit, remember, and when we drew our pictures of our cells way back in the first part of the year, we drew ribosomes as that little tiny dot, that little black dot. Okay, it was very small. It's not an organelle, but this is where we make our proteins. This is what makes everything that we see around us. So, on this ribosomal unit, in the large unit up here, we have two binding sites. Okay, your book doesn't name them, but it's the A site and the P site. And as the ribosome moves down the messenger RNA strand, we have our codons. Remember, this is a codon. Okay, any three letters in a row is a codon, and they have to match up. So when we went from DNA to messenger RNA, we did transcription. It's still nucleic acid language, okay? And then we were going to translate that here at the ribosome from nucleic acid language to our amino acid. And we see that on this structure over here. This is what our tRNA molecule looks like. It's still a single strand, but it has this weird form to it, like a key shape, which matches right to the A site and the P site. So at the bottom, and we make this really huge, make it really big, we have what's called the anticodon, which then has to match up to the codon of the messenger RNA. So what happens is the amino acid is bound to the P site. The P site promotes this whole thing. Okay? The ribosome is reading this direction. The ribosome is moving down the line right here. So it starts here. That's our start. Okay? As it goes down, it gets to this codon. These three letters have to match up to those three letters. So if these three letters were A, U, U, these three letters on my messenger codon have to be U, A, A. Remember our base pairing rules. For DNA, it was A to T. So if we put that up here really quick, DNA was A to T, G to C. When we did RNA, it was always A to U and G to C again. Our base pairing rules, just throw those up there really quick. Okay, so those have to match up. So this AUU is also attached to a certain amino acid. And this is where that circular model or circular table that you guys have in your guys' textbook comes into play. And on that circular table, you read from the inside out, from the bullseye, then you read yourself out till you got to the periphery where your amino acids are. The worksheet you're going to be getting, and I'll put this one up here, is one that's going to be square-shaped. Okay? You can definitely see the square-shaped. Okay? And on this one, it's the same thing except for we read the first base, the second base, the third base. Okay? So if we look at that on here, we look at AUU, that matches up to UAA. And the thing about all of our codes, all of these, whether it's square or circle, they're based on one RNA molecule only, and that is the messenger RNA unit. So if I want to know what AUU is actually coding for, I have to know what its messenger code is, UAA. So I look on here, it goes UAA, and actually codes for not even amino acid. This codes for a stop codon. It tells this to stop reading it. Okay? So you've practiced this, and we're going to do it again. I just set up a real quick chart here. So I have DNA, messenger RNA, tRNA, or transfer RNA, and the amino acid that it goes to. So the first one, I'm just going to start out with TAT. So what is the messenger codon that goes to that? Well, if I go to RNA language, I got to follow this code. So you can't have T always binds to what? A. A always binds to T, but if it's RNA, we can't have a T. It has to be a U, and this is an A. Okay? Then our tRNA, well, it's RNA language, so this has to be U, A, U. And if we look at this, what is UAU? Well, let's look up what is AUA. I'm going to use the square one, being it's the one in front of me. I go A, U, A. This is isoleucine. Okay? And on the circular one, it will come to the same one. And what you're noticing is that a lot of your amino acids are repeated. We have 64 possible codons and only 20 amino acids. So that's why we get that number. Remember, if we even go back a little bit further, we had 4 bases become 20 amino acids, become our 100,000 proteins. So that number that we started way last semester still keeps coming up. This little number line is super important. So let's start with this one. Let's do one where I give you the amino acid, but you have to work it backwards. So tryptophan means that I can figure out what my messenger RNA codon is by finding tryptophan. So I look on here and I find tryptophan, and I have to read it backwards. So it's going to be U, G, G. So this goes U, G, G. So now, what is the DNA code that goes to that? Well, U always has to bind to an A, C, C. So if it was my DNA inside my nucleus, I'd see ACC. For my tRNA, it would be A, C, C. Now, on this one, you can really see something here. The DNA can never leave the nucleus, but look, the message did get out. The messenger RNA transcribed it, but then the actual message is still out there, and we get ACC, which codes for tryptophan, which comes right off our DNA. The next one, I give us our messenger RNA codon, UGA. So I look at that one up, U, G, A. Oh, that codes for not an amino acid, but stop. So if this was reading a chunk of DNA, this is where it would say stop reading off this codon. Stop reading off these codons to make this amino acid or polypeptide chain. Okay, so we have A, C, U. And then up here, we go to our DNA codons. This has to be U. U always binds to an A, C, and this is T. Now again, ACT, ACU, almost the same language, but a little bit different. That's how we can get out of the nucleus. So as this is building up, as we start here, so this is where my amino acids are at. This one comes in and binds to it. It forms what's called the polypeptide, which we talked about also. This is called the peptide bond right here. Now I got to drink an arrow way out here. This is our peptide bond. This peptide bond keeps growing. This one shifts over as it reads. This one shifts over, kicks this guy out. This guy moves over. This one moves in. Okay, and they keep doing that over and over and over until they reach something that says stop. As soon as it tells it to stop, you have this little chain of amino acids. And then those go to the rough ER, they go to the Golgi bodies, and they get put together with other chains. And we get this big chunk of bubble gum that turns into some kind of structural molecule. Maybe it's part of your cell. Maybe it's part of the phospholipid bilayer. But that's that chunk of bubble gum, okay? And it all comes from these letters. So these 100,000 proteins are a result of being able to read this off as you guys work through this little problem. And this is one of the worksheets. Those of you that picked this up today, okay, this is Wednesday. You pick this up, you're going to be working on that. And then you have something that we're going to talk about here in a second called mutations. Okay, and I want to talk a little bit about mutations because there are mistakes. The polymerases that read this do only make that 1 in a billion mistakes, like I said before. That 1 in a billion mistakes does lead to some mutations. And we always have this connotation that mutations are bad, but we need to get that out of our kind of common psyche. So, really quick on mutations, and this will lead into the cancer part of your notes at the end. Mutations come in 2 types. There are point mutations, where we actually see on that list of codons. So we got A, C, U, G, U, A. Let's say this is a messenger RNA. Sometimes there's something gets substituted. As this was being read off, maybe this gets substituted with another U. Okay, that's a substitution. This may have changed what that codon is now. It may not have. Remember, the amino acids are repeated many times on those charts. So a point mutation may lead to an actual mutation. It may lead to no changes at all. The other two types of point mutations are when something comes in, say, I always use this as an example, you're out in the summertime catching some sun, and you're out there, and all of a sudden some UV light blows out one of your nitrogen bases, one of your codons. Okay, one of your nitrogen bases, sorry. So now that one's missing. We have to have what's called a shift because it can only read 3 at a time. So now we got CUG. Okay, what's that new codon? Maybe it codes for something to stop. Maybe it codes for a new amino acid. Maybe it's not isoleucine anymore. Maybe it's serine. Maybe it's adenine. Okay, it can do a lot of things. Then we have insertions. Sometimes we add in a letter. Oh, now we added an A in there. And I know this is kind of getting all kind of dirty and grumpy, but that still is going to, oh, now it shifted again. Okay, or it shifted it back this way, just depending on what's going on. That's called a frame shift mutation. And you can see that in your textbook. Your textbook does a great job on mutations, by the way, this new version of it. Okay, and then when we talked about chromosomes, we had 4 types. And again, this is why it's kind of a bummer we didn't get to have our classroom, because there's a lot of fun things that we can look at here on deletions. Because again, we have our classic chromosome, right? It's 2 chromosomes hooked together by their centromere. And then we can have deletions. In a chromosomal mutation, we're not seeing a single change in an amino acid. We might see, oh, this whole chunk just disappears, and then it reattaches, and it's shorter. That means we had a deletion. Sometimes you get a duplication. This happens when we know that these are homologous. Say this side breaks off, okay, and then it reattaches right here. So now we got the same genes being repeated again. Then this shows the example of another deletion. If I deleted that whole side, now this chromosome's missing a whole part of it, so that's another deletion. So a deletion and a duplication can happen. Then we have inversions. And this is something that actually is on my daughter's karyotype. She had a chunk of chromosome break off. And it was actually one of these upper ones. It broke off. So let's do this and let's put some marks on here. So this is what that looks like. Okay. Let's do something a little bit. Let's make this all dark up here. So that's what it looks like. So we got some shading on there. So in an inversion, it breaks off. It breaks off, and it reattaches upside down. Okay, that's a type of mutation. Does that lead to some kind of change? Not really. You know, normal female doing what normal teenage female girls do. But it's an inversion. Okay, if you get a karyotype done, you might find out if you have inversions on your chromosomes too. And then you can get a translocation. Let's say that breaks off there completely, and then you have one of your other sets of chromosomes. Let's say we have a little short guy over here. Not the same one. Let's get this up here. Let's say a chunk breaks off. Let's say this end part down here breaks off, and then it relocates itself way over here. That's called a translocation.
1 minute.
Okay. So those are going to end up with mutations. Now, mutations can lead to things like cancer, so make sure you read through that cancer section in your notes. It's pretty self-explanatory there. And the biggest thing I want you to get before my time's up here is that I want you to go away with that last sentence in your notes. Mutations are not necessarily bad. Mutations lead to changes, okay? And changes lead to evolution and variation. So we're all here as a result of mutations, okay? They're not always bad. They're also good. So hopefully that'll help out. This will be your last video.